Next, we're going to begin discussing how cancers develop within a cell. What are the processes that occur within the cell that lead to the development of uncontrolled cell division or cancer? There are 3 steps to this process: initiation, promotion, and progression. Cancer development is a multi-step, multi-feature physiological process. It does not happen all at once. So let's begin with initiation. A cell becomes initiated when it is mutated or epi-mutated in a particular way. So remember, mutation is a permanent change in the order of the bases in the DNA molecule, and an epimutation is a change in what's happening outside of the double helix that alters the expression of genes without changing the order of the bases. Once a cell becomes initiated, it moves into the second stage, which is called promotion. Promotion is the time when an initiated cell is somehow stimulated to divide. Now, promotion is a gradual process. Promotion is most of the time between an initiated cell becoming a cancerous cell. It requires prolonged, repeated exposure to a promoter substance. Now, a promoter substance doesn't have to be a carcinogenic chemical. It doesn't have to even be a foreign chemical. There are many natural compounds that act as promoters. For example, hormones. Estrogen is a great example. Estrogen is a hormone that stimulates cells to divide. We call it a promoter when it stimulates initiated cells to divide. There are growth factor chemicals too, whose job it is to stimulate cells to divide. They are stimulating growth in an organism. They become promoters when they stimulate initiated cells to divide. The third step in the road to cancer is called progression. Now, during progression, cells that have been initiated and then promoted to divide begin to acquire more and more mutations. These are mutations that create very strange, what we call aberrant traits. These cells become more and more aggressive in their characteristics. In other words, they're developing traits that we don't see in normal, differentiated cells. Now, what do I mean by differentiated? I mean a cell that has become fully mature. It has developed into a type of cell, a liver cell, a lung cell, and it is mature in that function. It is fully differentiated. Cells that move through this progression stage develop traits that we don't see in those differentiated cells. I have a list of the traits here. These are cells that develop a resistance to the immune system. They are able to evade certain cells that normally survey for damaged or altered cells and destroy them. Cells in progression are able to survive other mechanisms that would destroy them in the bloodstream. They develop the trait of being able to invade tissues, being able to spread into neighboring tissues. They are able to promote what's called angiogenesis, or the growth of new blood vessels. They sometimes de-differentiate. they take on traits of less mature cells. They show an increased resistance to various drugs that are normally cytotoxic, that normally kill living cells. And they show an increased resistance to the mechanisms of apoptosis, to normal programmed cell death. On this slide, you're looking at a diagram of how a cell becomes a cancer. You're looking at a diagram of initiation, promotion, and progression. I like this diagram because it draws out for us this idea that cancer is a multi-step process. Over on the left-hand side, you're looking at 3 cells lying side by side. This represents normal tissue. The cells are uniform size and shape. They're sitting next to each other in a nice ordered pattern. The nuclei are normally shaped and sized. This cell is going to be initiated. It's going to experience some kind of an issue with its DNA. Maybe it's going to experience a mutation, or maybe the DNA expression is going to change. And as a result, this happens. This cell, these cells, don't look the same anymore. They're trying to show us that they're just not normally growing at this point. They're no longer evenly sized and shaped. They're not in an organized pattern that we would see in normal tissue. They're then going to go through promotion. They're going to be stimulated to divide. And once they are stimulated to divide, or as they're stimulated to divide, they're going to be susceptible to more and more abnormalities developing. Remember, any time a cell divides, it has its entire DNA molecule replicated. And any time the DNA is copied, we run the risk of mutations. Imagine a cell that is being stimulated to divide. This is a cell that's dividing even more and even faster than normal, and so it is more likely to develop mutations. Now, the artist has changed the color of these cells to suggest to us that these cells are becoming more and more abnormal. The artist has drawn in some surface molecules. These might be proteins or glycoproteins or lipoproteins, and they're likely helping these cells to evade the immune system. And finally, a promoted cell is going to go through progression. It's going to gain more and more abnormal traits. It's going to be less and less and less like the original tissue that it emerged from, and it has now become a cancer. It's interesting to note that most of the research that helped us understand the stages of initiation, promotion, and progression, most of that work was done by studying the effects of carcinogens on cells. If you remember, though, carcinogens are not the only things that can lead to the development of cancer. Remember that certain infections can do it as well, as can ongoing chronic inflammation. Let's move on now to think about the role that certain infections play in the development of cancer. The history of this is actually quite fascinating. It was back in about 1911 that a scientist named Rouse, Peyton Rouse, first discovered that a type of cancer could be transmitted from one animal to another because of infection. Rouse discovered what would become called the Rouse Sarcoma virus. And this was actually a veterinary discovery because this is a virus that occurs in chickens. This was the first evidence that a viral infection could lead to cancer. Now, what Rouse did was he removed cancerous tissue. He removed this sarcoma that sometimes occurred in chickens. He took the tumors and he ground them up, as disgusting as it sounds, and mixed that cancerous tissue in with normal feed. And he fed it back to normal, healthy chickens. And what happened was some of those chickens that were fed the cancer tissue developed the same type of sarcoma. So what Rouse showed was that a viral infection that causes a tumor, a sarcoma in chickens, can in fact be transmitted from one chicken to another. Not only can the virus be spread, but the cancer can be spread because of the viral infection. RSV, or Rouse sarcoma virus, has been studied extensively since Rouse first did his experiments in chickens. And it's been shown to be linked to a particular type of gene called SRC. SRC, we pronounce it SRC. So this is a normal gene. This is a gene that normally occurs in chickens and other animals. It is called a non-receptor tyrosine kinase gene. So the protein that this gene codes for is an enzyme, and it's a kinase enzyme. It's a non-receptor type of tyrosine kinase. It phosphorylates the tyrosine amino acid inside of certain proteins. What we know now is that the Rouse sarcoma virus causes an infection that alters this gene. It alters this SRC gene, and it becomes what we now call an oncogene. Now, this onco, O-N-C-O, refers to cancer. You've probably heard that an oncologist is the type of physician that treats cancer. So an oncogene is a gene that is associated with cancer. Rouse sarcoma virus became known as an oncogenic virus, a virus that could initiate or cause a cancer. Now, how does it do that? Well, remember, it's either a mutation in the DNA or it's somehow an abnormal expression of the DNA. I want to mention that Peyton Rouse was awarded the Nobel Prize for his work with Rouse sarcoma virus. We now recognize that the Rouse sarcoma virus is one of many viruses that are oncogenic, capable of causing cancer. In fact, the current estimate is that up to 15% of all cancers are actually caused by a viral infection. Now, you've probably heard of a couple of these viruses already. You may know that, for example, certain forms of viral hepatitis can cause cancers. Hepatitis B and hepatitis C can cause cancers. HPV can cause cancers. Human papillomavirus has been linked to cervical cancer. We've recently started vaccinating young people against HPV. There are others that you may not have heard of. For example, the Epstein-Barr virus, which is one of the herpes viruses, has been linked to several different cancers, stomach cancer, certain oral cancers, and even a form of lymphoma called Hodgkin's lymphoma. There are other herpes viruses as well. There's one called herpes virus 8 that causes what's called Kaposi sarcoma in humans. We believe that we have only just located the tip of the iceberg when it comes to oncogenic viruses. It's likely that more and more of them will be discovered as time goes by. Now, it's not just viruses that have been linked to the development of cancer. There are other infectious agents as well. For example, there's a bacterium called Helicobacter pylori that has been linked to stomach cancer. There are certain types of flatworms that have been linked to liver cancers. Now, these are not the types of flatworms that, for example, Dr. Pelletieri works with. Those are free-living flatworms. I'm talking about parasitic flatworms. If you look at the image down at the bottom, I know it looks like this person is holding a leaf in their hand, but in reality, what they're holding is what's called a liver fluke. It's a flatworm. Look at the size of it. And it can infect the liver. This happens to be an infection in the liver of a cow, but there are parasitic flukes in most animals. Another organism that's been associated with cancer is an organism called Schistosoma, which has been linked with bladder cancer. So there are multiple types of infections that have been linked to cancers. The question then became, how does an infection lead to a cancer? Remember, it's all about mutation and epimutation. And we know now that there are certain genes, certain oncogenes that tend to be involved. I want to introduce another term here, and that's a proto-oncogene, with proto just meaning before. So there's a form of a gene that's referred to as a proto-oncogene before it becomes an oncogene. And we'll talk more about this in the upcoming slides. Don't forget, though, there's another mechanism in our list of the things that cause cancer, and that's ongoing chronic inflammation. We know that sometimes infections are ongoing. They become chronic, and associated with infection is inflammation. So let's talk a little bit more about oncogenes. We know that there are genes that are closely linked to the development of cancer. Any gene that codes for a protein that can stimulate the proliferation of cells, we're going to include in the list of proto-oncogenes. Remember, when we talked about the steps along the road to cancer, we said initiated cells are promoted to divide. Initiated cells have to be exposed to something that stimulates them to divide rapidly. These can be perfectly natural compounds like estrogen and like growth factors. So the genes that code for the compounds that can promote a cancer are called proto-oncogenes. The most important thing to remember about these is that these are perfectly normal genes, coding for perfectly normal proteins that do this perfectly normal function of stimulating cells to divide. It's just that sometimes they're stimulating abnormal cells to divide. They're stimulating initiated cells to divide. Now we're going to use the term oncogene to describe an abnormal form of a proto-oncogene, an abnormal form of a gene that has the potential to cause cancer. Now, it's tempting to think that oncogenes are just proto-oncogenes that have mutated. Sometimes they are. Sometimes a proto-oncogene becomes mutated and becomes an oncogene. But other times it becomes an oncogene because it's being expressed abnormally. So it's not that the base pairs have changed in their sequence. It's that this proto-oncogene is now being transcribed and translated abnormally. Remember, that's epimutation. At the bottom of all cancers, at the bottom of every single cancer, there is either mutation or epimutation. Since the time that Peyton Rouse did his work in 1911, over 200 oncogenes have been identified. Now, all of these abnormal forms of proto-oncogenes are producing proteins that fall into 6 categories. These are growth factor genes. These are transcription factor genes. And remember, we described a transcription factor as being any compound that's capable of binding to and interacting with the DNA molecule. These are proteins that are involved in the cell cycle, particularly in regulating apoptosis. These are receptor protein kinases and non-receptor protein kinases. I mentioned non-receptor tyrosine kinase earlier in the lecture. There are lots and lots of kinases, remember, and these are very powerful enzymes. These are enzymes that phosphorylate protein, and in the process, sometimes turn on proteins and sometimes turn off proteins. So they're very, very powerful. Finally, there is a group of proteins produced by oncogenes that are called regulatory Gtpases. These are enzymes that break GTP into GDP and phosphate and a little bit of energy. So these are the enzymes that break down GTP to supply some energy for different metabolic processes. You can imagine that these are very powerful enzymes. They're helping to provide the energy that cells need to do certain types of work. So 6 general categories of oncogenes. So how exactly does a proto-oncogene become an oncogene? How exactly does a gene, a perfectly normal gene, that makes a protein that normally stimulates normal cells to divide, how does it become an oncogene, a gene that somehow contributes to the development of cancer? There are 2 ways. The first way is that this proto-oncogene begins to make an abnormal protein, and there's only one way that can happen. We have to see a mutation in the proto-oncogene. And the second way is that we don't have abnormal protein, we have perfectly normal protein, but it's made in increased amounts. That is an alteration of gene expression. That's an epimutation. So sometimes proto-oncogenes, like all genes, simply get mutated. Something happens that causes the order of the bases to change. The order of the bases is what determines the order of the codons, and the order of the codons determines the order of the amino acids in the protein. Something changes to alter the bases, and we end up with an abnormal form of the protein. Now, when it relates to cancer, as it does with an oncogene, this abnormal protein, it's not that it's inactive. That's not the problem. The problem is typically that it's more active than it should be. These proteins are often enzymes, and they're out in the cell behaving with more activity than they should have. Sometimes it's just that that protein doesn't have any regulation anymore, so it's being active in a way that it shouldn't be. It should normally be regulated, it should be controlled in its activity, and through this mutation, it has lost that control. That's the mutation side of things. But remember, sometimes this is about epimutation. And through epimutation, we see increased amounts of a perfectly normal protein. Proteins are powerful things. And if a cell starts to produce a protein in excessive amounts, there can be problems, including the development of cancer. Sometimes this is through increased transcription. Sometimes this is through an increased stability of the messenger RNA molecule. It doesn't get degraded as quickly as it should, and so it is translated more than it should be. And sometimes this is due to strange mutations involving entire chromosomes. Remember, sometimes genes get duplicated. Sometimes the entire chromosome breaks apart and reassembles, and you get translocations. These are severe mutations that are not changing the order of the bases, but they're changing how much protein is being produced. If you duplicate a gene, you now have 2 copies of a normal gene. And that doubling of the gene is going to lead to a doubling of the protein made from that gene. If you break a piece off of a chromosome, it's not that you're changing the order of the bases, but you are taking certain genes out of their normal order, off of their normal chromosome sometimes, and putting them on another chromosome. And that can lead to a change in how they are expressed and a change in how much protein is made from them. So, infections can lead to cancer, and sometimes what's happening is the infection is causing proto-oncogenes to become oncogenes. Sometimes infections lead to cancer through our last mechanism, which is ongoing chronic inflammation. Now, I want to stress here the word chronic, because inflammation by itself is a perfectly natural process that does not lead to the development of cancer. It's perfectly natural for tissue to be inflamed when it's diseased or damaged. It's inflammation that helps the tissue to heal. It's a normal immune response. What's not normal is ongoing chronic inflammation, inflammation that does not resolve. Now, when do we see chronic inflammation? Well, I mentioned that chronic infections can do this. So infections that don't go away on their own, infections that don't get the right kind of treatment, they can become chronic, and they can lead to infections that last years and years. There are immune-mediated diseases that sometimes produce chronic inflammation, particularly if they go untreated. Things like rheumatoid arthritis, RA, or ulcerative colitis, UC. These are autoimmune diseases. The immune system fails to see that human tissue is normal. It sees it as foreign, and it tries to attack and destroy it. If it goes untreated, that creates this chronic state of inflammation. Finally, we see chronic inflammation anytime that tissue is chronically irritated. Now, by this, it might be a chemical exposure, perhaps in the workplace. It can be from particulate exposure. Remember the example of asbestos fibers sort of poking at the lung tissue like little needles. If these kind of exposures go on and on, they set up a chronic irritation and a chronic inflammation. There are lots of things that can create a chronic inflammatory process, including things I haven't listed here. The question is, how does chronic inflammation lead to cancer? If inflammation is a normal cellular and organismal process. The answer lies in what are called free radicals. We've mentioned free radicals before in this course. We said that a free radical is an atom that has lost an electron and is now traveling around with an unpaired electron in its valence shell. This atom is unstable. Remember, atoms seek to pair the electrons in their valence shell in order to gain stability. That's why atoms with unpaired electrons will often covalently bond with other atoms that have unpaired electrons. Sometimes atoms form with unpaired electrons and they become very dangerous inside the cell. They become free radicals because these atoms, rather than seeking out to form a covalent bond and share a pair of electrons, these atoms will seek to take electrons from other atoms. These free radicals will take electrons away from other atoms, causing them to become unstable. Free radical production inside of cells can ultimately lead to DNA damage because the DNA molecule can also be attacked by free radicals. And if the DNA molecule gets damaged in this way, it can lead to problems when the cell tries to replicate that DNA. It's very difficult for the DNA polymerase enzyme to copy DNA that has been damaged. And oftentimes what will happen is the enzyme will not know how to deal with the damage. It won't know how to read the bases in the damaged area, and it will place incorrect bases in the copy. So that leads to mutations. This is an example of DNA damage leading to mutation. In addition to these free radicals that can damage DNA, there are other chemicals that get released from immune cells during the inflammatory process, things like cytokines and chemicals called prostaglandins. These are pro-inflammatory chemicals, and they themselves can behave as promoter chemicals and progression chemicals. So if there are initiated cells in the area, and again, they may have become initiated by being exposed to free radicals, those cells can then go through promotion and progression by being exposed to ordinary, everyday inflammatory chemicals. And of course, any cell that goes through initiation and promotion and progression has the potential to become a cancer. Here's some important information. A single oncogene inside of a cell is usually not enough to create a cancer. That's good news for you and I. Chronic ongoing inflammation does not always create a cancer. That's good news for you and I. We recognize that cancer results from multiple mutations inside of a cell over time. And those multiple mutations must be followed up by promotion, remember stimulation to divide, and progression, further mutations, in order for a cancer to develop. It's actually kind of hard for a cancer to develop from a cell biology standpoint.